everyone, I am Shubhasri Pal. We are discussing this question from GATE Examination 2018, paper XLS, question number 3. And the question states, the oral polio vaccine OPV consists of A. Live attenuated virus, B. Killed virus, C. Viral toxin, and D. Viral capsid subunit. For more information on vaccination, you can follow this NPTEL course Immunology. And this lecture is active and passive immunity and vaccination. Now, coming to our discussion on the different types of vaccines. So, what is the difference between a live attenuated virus and a killed virus? What is it? Um, what is the difference between a toxin and a capsid subunit? So, let us discuss that a little bit. So, the different among the different types of vaccines that are available against different diseases, the most common is the live attenuated vaccine. The live attenuated vaccine are designed against diseases like measles, mumps, the Sabin vaccine of polio, the rotavirus vaccine, varicella, tuberculosis, and some of the advantages of live attenuated vaccine are the strong immune response. It can often produce lifelong immunity. So let us first discuss what is a live attenuated vaccine. Now, microorganisms can be attenuated or disabled so that they lose their ability to cause any disease. So that means they are attenuated or they are mutated in such a way that they lose their pathogenicity, but they retain their capacity for transient growth within an inoculated host. So if we inject a person with that microorganism will be able to grow in the host, but it will not cause any pathogenic symptom. So it will not cause any disease. Some agents are naturally attenuated by the virtue of their inability to cause disease in a given host. So we know that um, some the viruses they are specific for their hosts. So a particular type of virus um, cannot grow in or cannot infect all types of organisms, right? So they have their particular host. So that is how they can be attenuated naturally. So the first vaccine that was used by Jenner was of this type, that is the vaccinia virus cowpox. So inoculation of humans confers immunity to smallpox but does not cause smallpox. So this vaccinia virus actually causes smallpox in the cows, right? So that is why it was called cowpox. But this vaccinia virus can grow in human bodies, but it cannot cause the smallpox disease in human bodies. However, if we inoculate the humans with this vaccinia virus, then the humans they produce this immune response so that humans can be protected from the pox, right? Now, attenuation can often be achieved also by growing a pathogenic bacterium or virus for prolonged periods under abnormal culture conditions. This selects mutants that are better suited for growth in the abnormal culture condition than the human body or than the natural host. So, for example, an attenuated strain of Mycobacterium bovis, which is called the Bacillus calmette variant, the BCG vaccine, this was developed by growing the Mycobacterium bovis on a medium containing increasing concentrations of bile. Right, so this is not a natural condition for the virus to grow, but once we grow the virus in this abnormal condition for long and long time, so in that time, the virus will be attenuated to grow in that particular medium only, and it will not be able to infect the natural host. So that is how the BCG vaccine was developed. The oral polio vaccine, which is OPV, which was designed by Albert Sabin, it consists of three attenuated strains of the poliovirus. and it can be administered orally to the children. So this is the oral polio vaccine that we're talking about that was designed by Sabin, which is also called the Sabin's polio vaccine. The attenuated viruses colonize the intestine and induce production of secretory IgA, which is an important defense against naturally acquired poliovirus. So, this attenuated virus, they cannot cause the disease, but they come in the intestine of the host, and then they produce the secretory IgA molecule, Ig immunoglobulin A, which protects the individual against poliovirus. So this vaccine also induces IgM and IgG classes of antibody production, and ultimately it protects the person, the individual, from the three strains of virulent poliovirus. Right? So even though the vaccine that is uh, inoculated or that is injected in the host, that is attenuated strain. That is actually three attenuated strains of poliovirus, but so which is why they do not cause any disease in the host, but um, they protect the host against any further um, infection from poliovirus, from the virulent strains of poliovirus. Now, live attenuated vaccine was the very first type of vaccine that was developed, designed. There are many advantages of the live attenuated vaccine, like they are more immunogenic. They are very efficient in production of memory cells because they are growing inside the host. So their immunogenicity is high. They can cause production of memory cells, and they often require single immunization. So a single immunization will be enough for them to produce or provide lifelong immunity. But there is one major disadvantage of live attenuated vaccine, which is they can mutate and they can revert to the virulent form. So if they revert to the virulent form, what is the consequence? The consequence is that it can cause the disease, right? So for example, the polio virus, if it reverts to the virulent form inside the host, then it will cause the polio disease in the host. So, which is a major disadvantage of the live um, attenuated vaccine, which is it may mutate to virulent form. The next type of vaccine that we will talk about is the inactivated vaccine or killed vaccine. So basically, the virus is inactivated or killed to produce this kind of vaccine. Some of the examples are cholera vaccine, influenza vaccine, and the Salk vaccine of polio. Okay. Now, what is inactivated? How is it different from the live attenuated virus? So, the inactivated vaccine is the virus is inactivated by treatment with heat or chemicals, right? So the virus is the virus or the pathogen that is killed. The so this pathogen is killed. It's not live anymore, and it cannot replicate anymore, right? So it is incapable of replication, but it still allows to induce an immune response to at least some of the antigens contained within the organism, right? So therefore, the virus or the pathogen is killed. It is inactivated. It cannot replicate inside the host. It is not live anymore, but it can cause the immune system to provide immunity against this type of pathogen. So it can induce the immune system to provide immunity against some of the antigens contained within the organism. And it is very important to maintain the structure of the epitopes on the surface of antigens during this inactivation, because the epitopes are what are recognized by the by the immune system, right? The epitopes are recognized by the antibodies. So it is very important that during inactivation, the structure of these epitopes are not destroyed. Heat inactivation is often unsatisfactory because it causes extensive denaturation of the proteins and therefore it causes destruction of the epitopes. So, for example, epitopes that are higher orders of protein structures, like the tertiary structure of protein, they are likely to be altered significantly because of heat denaturation. Chemical inactivation with formaldehyde can be used, or other alkylating agents can be used. The Salk polio vaccine is produced by formaldehyde inactivation of the poliovirus. Now, the Salk polio vaccine is a better vaccine than the Sabin polio vaccine. Why is it called? Because the Salk polio vaccine is an inactivated virus. And since it is inactivated, it cannot revert to its virulent form inside the host, right? So, therefore, it has no chances of causing the disease. But since this is an inactivated vaccine and it cannot replicate inside the host, so therefore it primarily produces humoral antibody response. And since it is an inactivated vaccine, it requires repeated boosters, um, unlike the single immunization of live attenuated vaccine, right? So these are some of the advantages and disadvantages of the inactivated or killed vaccines. So it is stable, it is safer than live attenuated vaccines, but it produces weaker immune response and it requires booster shots. So the oral polio vaccine that is asked in this question, that is basically a live attenuated type of virus. The oral polio vaccine, which is designed by Sabin. So it is a live attenuated virus. So the answer here will be live attenuated virus. But the Sabin polio, this is the Sabin polio vaccine, and the Salk polio vaccine is inactivated type of virus. So where uh, the virus is treated with formaldehyde to inactivate the virus. Now I would like to discuss a little bit about the other types of vaccines as well. For example, um, there are some purified macromolecules that can be also used for producing vaccines like toxoid, subunit vaccine, and conjugate vaccine. So first of all, let us talk about the subunit vaccine. So there are many risks with attenuated or killed whole organism, as we were talking about, and these risks can be avoided by a strategy which uses only specific purified macromolecules which are derived from the pathogen. So we will not heat inactivate or chemically inactivate the virus, or we will also not attenuate the pathogen or the virus. Rather, we will use a purified macromolecule from the pathogen as a vaccine. So that is subunit vaccine. There are three most common applications of subunit vaccine. One is the inactivated exotoxins or toxoids, the capsular polysaccharides or surface glycoproteins, and key recombinant protein antigens. So, recombinant protein antigens, polysaccharide antigens, or toxoids. So, we will first talk about toxoids. Toxoids are bacterial pathogens like, for example, the bacteria that causes diphtheria and tetanus, right? Diphtheria is Corynebacterium diphtheri and tetanus also. Uh, so these bacterial pathogens, they produce exotoxins that cause the disease symptoms. And once these bacterial species, they infect their host, the bacteria produces this kind of exotoxins which are responsible for the disease symptoms. Now, diphtheria and tetanus vaccines have been made by purifying this particular exotoxin from the bacteria. So, purifying the bacterial exotoxin and then inactivating it with formaldehyde. to form a toxoid. So it's not a toxin. So what is the difference between a toxin and a toxoid? A toxin is a compound that causes the disease, that is that comes from the bacteria when it infects us, right? And toxoid is the inactivated toxin, right? So using formaldehyde, we can inactivate this bacterial exotoxin, and that forms a toxoid. Now, once we vaccinate with this toxoid, it induces antitoxoid antibodies. Of course, um, it is an antigen. So once it goes into our body, our body will produce antitoxoid antibodies, which will be capable to bind to the toxin and neutralize the toxin. So there will be no side effects, no disease symptoms, but it will produce immunity for this toxin. Now, coming to the subunit vaccine. So, toxoid examples are diphtheria and tetanus. And advantage is that the immune system becomes primed to recognize these bacterial toxins. Coming to subunit vaccine, actually the virulence of some pathogenic bacteria depends on the antiphagocytic properties of their hydrophilic polysaccharide capsule. So they have a polysaccharide capsule around them, which can evade phagocytosis by the immune cells. So coating the capsule with antibodies or or complement greatly increases the ability of macrophages and neutrophils to phagocytose those pathogens. So this particular capsule has some properties which can evade phagocytosis. So it prevents phagocytosis by the immune cells. But once we coat this capsule with some antibodies or complement proteins, then the immune cells will be able to recognize this capsule, and it will be able to phagocytose this kind of pathogens. Now, these findings have led to the development of the vaccine, which consists of purified capsular polysaccharides, right? So, capsular polysaccharides are purified, which are actually the part of the capsule of the um, pathogenic bacteria, right? So purified capsular polysaccharides, and these capsular polysaccharides are used as vaccines. So once we introduce these antigens inside the body, or once we introduce these capsular polysaccharides as vaccine into the body of an individual, then the individual will produce antibodies against those polysaccharides, right? And once a real infection occurs, so for example, this pathogenic bacteria invades the host body, then uh, there will be antibodies produced inside the individual. These antibodies against the polysaccharide on the capsule, right? So there will be antibodies host um, in the host's body, which will recognize this capsular polysaccharides, right? And therefore, these antibodies will coat the capsule. And once this capsule is coated by those antibodies, the macrophage and the neutrophils can easily um, recognize them and phagocytose them, right? So the current vaccine for Streptococcus pneumoniae, uh, the organism that causes pneumococcal pneumonia, consists of 13 antigenically distinct capsular polysaccharides, PCV13. Right, so the pneumonia vaccine that is used currently is capsular polysaccharide vaccine, which is a subunit vaccine. And the last one, so it is also the specific antigens lower the chance of any adverse reactions, right? Because it is only a polysaccharide that is introduced in our body, right? So it should not cause any adverse reactions, but it is difficult to develop. And last one is the conjugate vaccine. So conjugate vaccine, oh, I have not written it here. Conjugate vaccines are a conjugate of two types of pathogens, and it primes the infant immune system to recognize certain bacteria. Now, I will also like to talk about the recombinant vector vaccine. The recombinant vector vaccine. So, live attenuated vaccines They provide antigen delivery. Um, the live attenuated vaccines, they prolong the antigen delivery and encourage cell-mediated responses. So what do I mean by prolong the antigen delivery? So since the live attenuated vaccines, they are live and they can replicate inside the host. So they continue replicating inside the host and they or are always providing the antigen in the host system so that the host can recognize and produce antibodies. Right? So that means it has a prolonged antigen delivery period and encourage cell-mediated responses as well because the live attenuated vaccine or the pathogen is present inside the host, so cell-mediated responses will be triggered. But the disadvantage is that they can sometimes revert to pathogenic forms, as I mentioned. Recombinant vectors maintain the advantages of live attenuated vaccines but avoid this major disadvantage. The individual genes which encode these antigens of especially virulent pathogens can be introduced into these attenuated viruses or bacteria. So basically, the virus or the bacteria that is engineered, that is made recombinant with genes for virulent pathogens. Now, the attenuated organism now serves as a vector, which is why it is called a recombinant vector vaccine. And this vector can replicate inside the vaccinated host. So this vector is used as a vaccine, and this vector can replicate inside the host. So of course, it will prolong the antigen delivery as the live attenuated vaccines, and it will express the gene product of the pathogen. So the pathogenic gene that is cloned in these recombinant vectors, so that will actually cause the expression of the gene product of the pathogen. And this will be recognized by our immune system, and the immune system will provide um, immune response against that particular gene, pathogenic gene, or the virulent, the key antigen of the virulent pathogen. Right? So that is the most important advantage of recombinant vector vaccine that it can replicate inside the host, it can prolong the antigen delivery, it will also mediate cell-mediated responses, it will also induce cell-mediated responses. But since most of the genome of the pathogen is missing, reversion potential is virtually eliminated. So it should not revert like the live attenuated vaccines, right? So theoretically, it should not revert back to any virulent form because most of the genes of the pathogen is actually deleted. Only the gene that produces a key antigen that is only present in this recombinant vector. So, of course, the advantage of that is that it mimics natural infection and results in strong immune response. So, these vector vaccines are now currently in under development. And the other one is the DNA vaccine. Now, DNA vaccine is a more recent vaccination strategy, and it utilizes plasmid DNA encoding the antigenic proteins that are injected directly into the muscle of the recipient. So, what is the difference between a recombinant vector vaccine and a DNA vaccine? In a recombinant vector vaccine, we are using a attenuated virus or bacteria as a vector. But here, in case of DNA vaccine, we are not using any vector. So we are using a plasmid DNA, which has this antigenic protein cloned into it, and it is directly injected into the intramuscular injection in the muscle of the recipient. And this strategy relies on the host cell to take up the DNA and produce the immunogenic protein in vivo, right? So this DNA will be taken up by the host cell and it will produce the immunogenic protein. And therefore, what will happen? It will these proteins will be produced inside the cells, right? So it will direct the antigen through the endogenous MHC class 1 presentation pathways. So since the protein will be present inside the cell, it will be directed by the MHC class 1 antigen presentation pathway and since it is associated with MHC class 1 pathway, so it will activate better cytotoxic T helper cells, cytotoxic T lymphocyte responses. Right, so cytotoxic T cells, they will be better adapted to recognize these kind of protein antigens. And the DNA appears to either integrate into the chromosomal DNA or be maintained for long periods as an episome. Okay, so either it will be integrated or it will remain separate from the chromosome and is often taken up by the dendritic cells or muscle cells in the injection area. So it's a foreign DNA, so it will be taken up by the dendritic cells or the muscle cells in the injection area, and then further processing will occur through the MHC class 1 pathway, and it will give us a better cytotoxic T cell response. So CD8 plus T cells response will be better. And these are some of the infectious diseases against which the DNA vaccines are already being developed, like the HIV, influenza, malaria, hepatitis B. I would also like to mention that the current pandemic, which occurred 2, 3 years back, the COVID-19 pandemic, and the vaccines that were developed to tackle the COVID-19 pandemic, one of them, or I would say a few of them, were RNA vaccines. So, like the Moderna vaccine, the Pfizer Moderna vaccine, that was the RNA vaccine. So, it was an RNA that was uh, actually introduced into the cells of the recipient, right? And that was producing immunity against the COVID virus. So that is all about the different types of vaccines. And we have already discussed the answer of this particular question, that the oral polio vaccine consists of live attenuated virus. And that's all. Thank you.